welcome everyone for joining online. Okay, so I'm delighted to introduce Manoj Patel, who's Associate Professor of Anesthesiology at the University of Virginia School of Medicine. So Manoj completed his BSc and Ph.D. in Physiology at the University of Coventry in the UK, and then he took on postdoc positions at the University of Virginia, and then back to the UK at University of Bristol, followed by a joint position between the University of Cambridge and Pfizer UK. Manoj then returned to the University of Virginia in 2000, where he set up his own group. So over the past 20 years, Manoj's lab has investigated the role of voltage-gated ion channels in the development of epilepsy, with a focus on sodium channels and T-type calcium channels and their role in controlling neuronal excitability. And so in particular, the relationship between these ion channels and neuronal hyperexcitability associated with epileptic seizures. Okay, not only has he been busy in the lab, but he's also founder and chief scientific officer for the biotech company GenEp, which is focusing on sodium channels as therapeutic targets for epilepsy. Okay, so Manoj, if you're ready, then yeah, then you can fire away.
So good morning and good afternoon, depending on where you are. And thank you, Eddie and Nick, for the invitation. My name is Manoj Patel. I'm at the University of Virginia, and this is if you can see my screen okay?
Yep, all looks good.
Great, thank you. This is a beautiful picture of the University of Virginia where I'm located. So the title of my talk is Gain-of-Function Mutations in NAV 1.6 and How They Lead to Epileptic Seizures. Disclosure, I am the CSO for GenEp. And the outline of my talk is going to be, first of all, I want to introduce what SCN8A epileptic encephalopathy really is. And that is, SCN8A is the gene for the NAV 1.6 sodium channel isoform. And it's expressed in both excitatory and inhibitory neurons. And there are patients that have mutations in this gene that lead to epilepsy. Then I want to touch a little bit on the importance of the NAV 1.6 isoform in temporal lobe epilepsy. So this is a much larger population, of which about a third of these patients are therapy resistant. So there's really very little treatment options for these patients. And so any kind of drive towards better molecules is much required right now. And then finally, I'll just tell you a little update on what's out there for targeting 1.6, not only for the treatment of this orphan disease, the SCN8A, but also for generalized epilepsy. So if you go back to your university days, you saw an action potential, which looks a little bit like this. And the reason I show you this is just to emphasize how important sodium channels are in controlling the excitability of neurons, both excitatory and inhibitory. So there is evidence that sodium channels are open at subthreshold potentials that kind of enhance the excitation coming from synaptic transmission. They're clearly, particularly 1.6, is involved in driving the neuron to threshold. And once you reach threshold, we know that they play a very important role in driving the depolarization and the upstroke. And then, of course, you have potassium channels, which are also very important in repolarizing. So you can imagine that any kind of disruption to the way that this sodium channel, particularly 1.6, behaves is going to have a fundamental effect on the excitability of neurons. So there are many sodium channel isoforms, at least 9. And the ones I'm going to be talking about today are the central nervous system express sodium channel isoforms. So NAV 1.1, 1.2, and 1.3. And 1.6. And this is an old slide, so you can see that we have inherited diseases for 1.1 and 1.2. But we know there are patients that have mutations in the NAV 1.3 isoform that give you epilepsy. And today I'm going to tell you a lot more about the patients that are out there that have gain-of-function mutations in NAV 1.6 that give you epilepsy. Now, from a biotech perspective, the one channel you want to try and avoid is the cardiac channel, the 1.5 channel, because obviously if you have compounds or drugs that affect that channel, you have the risk of developing cardiac abnormalities. So the sodium channel itself looks a little bit like this figure from a very old Cattrall review. It's a voltage sensing. That means that when the voltage changes within the cell, there is a conformational change in that channel protein, allows sodium ions to flood in. It consists of 4 homologous domains. There's domain 1, 2, 3, and 4. The transmembrane sections, there are 6 of them, and the first 4 are thought to be the voltage sensor, and the fourth one in particular has a lot of charge residues that basically sense the voltage in the cell, and they move when there's a change in voltage, and that allows the channel to open. The transmembranes 5 and 6 tend to line the pore, so a lot of the clinically used anticonvulsant drugs that are out there bind to residues within the S5 and S6 of the pore. What's important with sodium channels is that they have this fast inactivation gate, which is the 3,4 linker, which is heavily involved in inactivating the channel. Now, the channel itself has either 1 or 4 different beta subunits that can bind to it, and the beta subunits are very important in controlling or fine-tuning the activity of the channel, and also helping the channel get traffic to the membrane. So 1.6 is heavily expressed in the central nervous system, particularly the nodes of Ranvier and the axon initial segment. So the nodes of Ranvier are these areas of unmyelinated axons where you get saltatory conduction as the action potential kind of jumps down. And the reason that happens is because you've got this regenerative current, mainly from NAV1.6, that these channels are expressed at these nodes. And here's an image of red staining that shows a node of Ranvier, and you can see that the red staining is the sodium channel. So they're highly concentrated at these nodes. And this image is from my lab, which shows an axon initial segment. Here, the green is the sodium channel expression, and anchorin G is a marker for the axon initial segment. So you can see there's a lot of expression of 1.6 there. And so these channels are very important because they have a lower AP threshold, allows the isoform to contribute to what we call burst firing or repetitive firing and neuronal excitability. So normal function of the sodium channel at rest, sodium ions cannot flow through the channel because the channel's in a closed state. And what you can see is that the inactivation gate shown in this cartoon is open, but obviously the channel is not open. When there's some kind of depolarization, there's a conformational change in the channel. And what you see now is that the sodium ions are coming into the channel. And if you're an electrophysiologist like me, you'll recognize this trace at the bottom, and that's really showing you the inward current of sodium that is coming into the cell. But with time, even if you're still depolarizing the cell, the channel will inactivate. And so the idea is for fast inactivation, this gate swings in, occludes the pore, and prevents sodium from coming into the cell. And that's our normal function in sodium channel. Now we know that there are lots of disruptions to sodium channels that lead to disease. And the disease that I'm particularly interested in is epilepsy. And so epilepsy is really, if you like, it's like a storm in the brain. It's overexcitation. These neurons continue to fire, rapid action potentials, leading kind of complete overexcitation and a seizure. And it depends on where that really starts and where it progresses to. That's the clinical phenotype, if you like. So most patients start off in one area of the brain, and it can spread to another area and spread towards the whole of the brain. And that's normally what we call generalized seizures. So epilepsy is a serious neurological disorder. It affects about 1 in 26 people just in the US, over 3 million people. It's the 4th most common neurological disorder in the US, and many people every year are diagnosed with it. It's a factor in the death of a lot of people too. So, you know, they die from car accidents or drownings, and they don't know when these seizures are going to happen. So it's a very serious disorder and it contributes to a number of deaths that are not related to dying from the actual seizure, which is known as SUDEP, sudden unexpected death in epilepsy. Unfortunately, there is no cure for epilepsy. Seizures can only be treated pharmacologically to stop them. And this is where the problem lies, because about 30% of epilepsy patients are refractory to current meds. They're usually on more than one, and there are serious side effects associated with the meds that are currently out there. And the only other option for these patients is surgery, where those who are lucky to have a foci that can be resected undergo this type of surgery. But even in those patients, they typically do not take them off anticonvulsant drugs. That's just too much of a risk. And I'm interested in these mutations. So about 5% of patients now have a cause for their epilepsy, and they typically have mutations in sodium channel genes. SCN1A is Gervais syndromes, typically a loss of function in NAV1.1, or as I'm interested in 8A, and there's also 2A, where there's a gain of function and loss of function in 2A that can lead to seizures. So for the last 25 years, we've been very interested in understanding what happens to sodium channels in not only genetic epilepsy, but in acquired epilepsy. So they are people who've had head traumas or other viral infections that lead to epilepsy. What happens to the sodium channel? Because neurons have to become hyperexcitable somehow to allow you to have a seizure. So how can we better understand what goes on, and also how can we treat them? So I want to give you a little bit of history about the patient with SCN8A epileptic encephalopathy, and this is a picture of Shay. She had unexplained refractory epilepsy at 6 months of age, epileptic spasms at 4 years of age, and the spasms are kind of like little jerks that these patients tend to have. She didn't walk till she was 3, spoke very little words before 4 years. She had uncontrollable seizures that led to intellectual disability. So a lot of these patients, they lose neurons when they have seizures. If they have a whole bout of these seizures, they lose a lot of neurons, and therefore any progression in speech or motor function is typically lost. Developmental delays, hypertonia, balance problems, and their vocabulary is very limited. With Shay, because we didn't know about SCN8A gain-of-function mutations, they thought she was autistic at 5 years of age. She had a lot of seizures, 50 to 60 a day at the age of 5. Her MRI was normal, and unfortunately she died at the age of 15 in her sleep after having a few seizures the day before. What's interesting about Shay is her father is Michael Hammer, and he's a research scientist in Arizona. He's an evolutionary biologist. And he decided to have some of his lab try to figure out why Shay was having these mutations, and gave the project to a talented postdoc, Krishna. And it was about 2 months after Shay died that they finally discovered this gain of function, or this mutation. They didn't know what the significance of the mutation was, but they found a mutation in SCN8A. And it turns out that this particular one was right here at the end of the transmembrane domain S6 of D4, and it's right where this cytoplasmic tail appears, if you like. And we know that the cytoplasmic tail and the inactivation gate can kind of come together and control inactivation of the channel. So Michael did a Google search and found Professor Miriam Meisler at the University of Virginia and contacted her, and she said, yes, we work on sodium channels, and they made the mutation, and this was Hersheys mutation. It was evolutionary conserved through all other vertebrates and invertebrates, so clearly this was an anomaly for her. It was an asparagine to aspartate. They also got in touch with Steve Waxman at Yale, and between the three of them, they made the mutation, expressed it in cells, and they did a bunch of electrophysiology or biophysical characterization. And one thing they noticed was this increase in the persistent sodium current. So here in this example, this is a wild type channel, and you can see there's a little bit of current. It's about 1% of the peak current. It's very small. But in Shay's mutation, it was much larger, and you can see it's almost 20%. Another interesting factor is the fact that in these cells, they saw a reduction in the current density, but that just could have been an artifact of the cell system they were using. But the bottom line is they showed this large persistent current. And I use the analogy of if I use English terms here, a dripping tap. So you have a faucet or a tap and it's dripping. And if the sink is blocked, eventually that sink is going to fill and it's going to flow over the sink and flood your bathroom. So the idea is that you've just got this consistent leaking sodium current that's depolarizing, and that's going to drive that neuron to threshold much more easily, and eventually you're going to have a network that's hyperexcitable, and that's going to give you the seizures. So what Shay did do for all the other families is provide a target, if you like. So now SCN8A is on the epilepsy screening panel, along with other ion channels and ligand-gated ion channels as well. So if a patient comes into the clinic and has had a suspected seizure, they're most likely going to be tested to see if they have a mutation in 8A. And here is a cartoon of just some of the mutations that have been discovered. We have over 450 of them just in the US, but we recently found out there's a doctor in China who has 150 cases just in his region. So this is going to explode big time, because now that every single patient that comes in is being tested, we will find more and more of them. And because this channel plays such a crucial role in controlling excitability, you can imagine that its mutation is going to be devastating. So some of these mutations are located in areas that we kind of make sense, like the S4 or the inactivation gate or the C-terminal tail. But other mutations are located in areas where it's really unclear why that would lead to a gain of function. And we're talking about gain of function here, so overexcited channel that doesn't fully inactivate. And here is a picture of some of the families that I'm involved with, and they're part of this group called the CUTE syndrome. And we meet every year at the American Epilepsy Society meeting, and they do bake sales and 5K runs and things to raise money for research. So they're a lovely group of parents who really care for their children and are really hoping for some kind of cure. About a third of these mutations are recurrent. They happen, they're de novo, so their siblings don't have these mutations. This particular child somewhere during embryonic development had a mutation, and they're the only one in the family that has this problem. So they're recurrent at some of these sites because that's where we think DNA methylation occurs, and there may be some disruption during embryonic development. Most patients have seizure onset about 4 months of age, but it can be between 4 and 12. There is evidence of mothers coming into the clinic and complaining about a rumbling in their tummy, and we believe that the fetus is actually having a seizure. So it turns out that, as you can see from this cartoon, there are mutations located all over the channel. So depending on where these mutations are, how devastating the effect of that mutation on the biophysical property is, that's the type of patient that is diagnosed much earlier. So someone who's a month old or has a seizure during birth most likely has a mutation in a very critical part of the channel, whereas someone who gets diagnosed 12 months later, first seizure, they're 1 year old, they're likely to have a mutation that's causing less of a severe change in the biophysical properties of the channel. So that kind of makes a lot of sense to us. So they have a lot of other issues, comorbidity issues, cognitive disabilities. The biggest concern is SUDEP. About 10% of these patients will have are expected to have SUDEP, which is sudden unexpected death in epilepsy. This can rise to 40% if their seizures are not being controlled. So what really happens is this beautiful cartoon from a cardiac channel review for long QT3 syndrome, but what you can see is somehow the channel is not completely inactivated anymore, and you get this leaking of the sodium current, which gives you this persistent current when it should really be where this black line is. And that is what's causing these seizures to occur. I told you there are recurrent SCN8A mutations, and 2 of them are the 81617 site and the 1872. We now have a mouse model for this mutation, which is wonderful. So we're now able to test this particular mutation now. It's Cre dependent, so we can express it in various neurons to see the mechanism by which specific neurons play a role. This is an old slide. We now know there are over 50 cases within the US of this particular residue, and I think 40 of this 1617. So there are these hotspots that occur because we think that's just where DNA methylation occurs. What we find with all of these is that they do have a disruption in the way that the channel inactivates. So here's a wild type channel and here's a mutant channel, and you can see for the 1872 mutation it's the same, and you can see that the wild type decays at a certain rate, whereas the mutant channels don't. And so that is this disruption to the way that the channel inactivates. What we also find, something that's quite interesting, is that within a group of patients that have the same mutation, their seizures can be a little bit different. So here's an example of 5 patients that have this 1617 mutation. You can see patient 1 was age of onset was 3 months, whereas patient 5 was 12 months. They all have this. They all have seizures, so generalized tonic-clonic seizures. But you can see that drug response is different. So carbamazepam for this particular patient was somewhat effective, but here this patient is totally refractory. So again, we're a genetic mix, and therefore there are other genes that can be somewhat protective. And so this is an interesting area that a lot of geneticists are focusing on. Why are so many of these patients so different? But they all have very serious seizures, so that's what we are interested in, is trying to control those seizures. So if we go back to Shay and Miriam, Professor Meisler, Miriam made a mouse model. She put Shay's mutation in the mouse, and if you have 2 copies of this mutation, the mice die at 3 weeks of age very quickly of multiple seizures. If you have 1 copy, the mice die. They start to have seizures around 6 to 7 weeks. The 2 copy mice, the DDs, the homozygotes, will have seizures at 3 weeks of age, and that's because the expression profile in mice and in humans, we believe, increases from birth. So by 3 weeks of age in mice, it's completely replacing 1.2, which is initially expressed at the axon initial segment and at the nose of Rombier. So once 1.6 starts to take over, you can see that the seizure onset begins, and then the seizures fully develop by that time. We have this mouse, and we actually have about a 97% penetrance. Miriam has a 50%, and we're not quite sure why that is. But our mice will almost all of them have seizures, and almost all of them will die. It's very few that don't have that phenotype. So I want to show you a video of a patient. The reason I say that I want to show you this video is because in any kind of disease, particularly if you're going to use it to screen for therapy, you have to have a very good clinical model. And I believe our SUDEP mouse model, or our mice model of SCN8A, is very good. So here's a video, and hopefully you can see this video. This is a patient in the UVA ICU unit that came in with suspected seizures, and they're monitoring this particular person's seizures. The nurse actually thinks that the patient's fussing at first, but what's really happening is that the patient is actually having a seizure. So they realize that quite quickly, and they try to make the patient comfortable. And what you'll see is this particular part right now, and that is the patient is going to go into what they call a tonic seizure. So right now, that this patient, all the patient's muscles are contracted. The patient is not breathing. The heart rate is increasing in tachycardia. So nurse says no muscle tone, no apparent breathing, and if we had the sound on, you'd see that the oxygen sats would be dropping. Now, I would tell you this child is now about 11 years old and is somewhat controlled. The seizures are somewhat controlled with sodium channel blockers that are clinically available, so that this particular one patient is doing quite well. But if you watch my mouse, and you'll see that when the mouse goes into a seizure, it does more or less the same thing. So this is this critical tonic phase. So right now the mouse is not breathing, all the muscles are contracted, but as the mouse comes out of this seizure and it becomes more of a clonic seizure, you'll see the limbs start to kick. And in the patient video, had I continued that video, you would have seen the patient start to gasp for air, just as this mouse is doing, and move muscle. So again, the reason I show you this is because we just have this really, really highly clinically relevant mouse model that we can study the mechanisms of these seizures, but also test novel pharmaceuticals. So we were very lucky that Miriam Meisler is very generous and shares the mice with people, and she shared the mice with us. And because we're electrophysiologists, we decided to first make brain slices from these mice and see what's going on at the neuronal level. And we did this at 3 weeks of age. So just to be clear, these D+ mice that have one copy of the mutation are not going to seize for another month at least. Whereas these DD mice, we did it at 3 weeks of age, are seizing and about to die any day. In fact, many of these mice had a seizure when we brought them to the lab to remove the brains. And what you can see is in wild type, if you inject current to polarize the cell and generate an action potential, and that's what these spikes are, they're action potentials, you can see you get no firing at, say, this particular current injection, but in these mice that have the gain of function mutation, whether it's one copy or two copies, you see that they fire a lot more action potential. So they're hyper excitable. You'll also notice that the action potentials look very different. They're very wide. And although they don't come up and go back down, we believe that that would encourage passive current movement or depolarization and allow for synaptic transmission to occur at a much higher rate, generating seizures. We also stimulated them synaptically, and what we found is that, and this was all done in the entorhinal cortex, which is a big input to the hippocampus, an area that's very important for seizure progression to the cortex. When we stimulate them, wild-type neurons fire a single action potential, but if you increase that intensity twofold or threefold, they continue to just fire single action potentials. That's in these neurons. Now, you could make other neurons within the brain burst because that's what they do, but in these neurons, they don't burst at all. But in our mice, the same neuron did burst, and you can see they become very bursty and what we call epileptic-like. So this is what we would classify as an epileptic form activity. So lots of bursting action potentials, which would drive synaptic transmission. Another current that we're really interested in is this current known as the Resurgent Current. And it's a current that kind of comes on as the cell repolarizes, and that's because when the sodium channel opens, a peptide from the beta-4 subunit, the tail of the beta-4 subunit, will occlude the pore and prevent the inactivation gate from binding and inactivating the channel. However, as the cell repolarizes, as it comes down again, this block is removed and the channel can open and sodium ions can pass through. And so you end up getting this inward current as the cell repolarizes. This particular current is really important because it drives threshold for the next action potential. So the larger the current, the more likely that you're going to get more action potentials generated, because you're going to provide a more depolarizing interspike current, if you like. And we looked at these currents, and we also looked at ramp currents, which again is another way of looking at these non-inactivating channels. And what we found is that they were larger in these D+ mice that were still a month away from seizing, and much larger in the mice that were actually seizing that moment. And we believe that this is the progression. And we've got some preliminary data where we recorded the same currents at 9 weeks of age in mice that have one copy but are seizing, and these currents are really large. So one thought is that once these currents develop into to a certain degree, or in terms of amplitude and gating, that's when the seizures really kick off. And you can see that this persistent current that we record using a ramp is also much larger in the D+ and larger still in the homozygote mice. So we also looked at just the activity of these neurons. We did outside-out patches. So we patched onto these neurons and recorded the currents. And what we found was that there was significant delay in the decay of the macroscopic currents, and also an increase in the persistent current, just that they had seen in the cell lines, but we did not see any difference in the current density. So they have the same current amplitude, if you like. It's just that they just don't inactivate, so the current is on a lot longer. So here's the persistent current in these neurons. When we looked at inactivation gating, they had disrupted, which means that these channels just don't inactivate. They're available for longer. And because activation was normal in these neurons, what you really end up getting is this difference in what we call the window current. So that's where the activation and inactivation overlap, and it gives you this range at which the voltage for these channels to be available to open and be able to open. So again, this is all encouraging neurons to become hyperexcitable. So there is a big drive to provide or to develop 1.6 selective blockers for this disease, and also for temporal lobe epilepsy. And what we found was we didn't use a 1.6 selective blocker, but we used very low TTX. We used 50 nanomolar TTX. And when you use a low concentration, you kind of just titrate down the sodium current available. And what we found is that in wild type, it really didn't have much of an effect. But when you look at the bursting that we see in the D+, the hets, and the homozygote mice, you can see the bursting. TTX was able to somewhat normalize it. So again, this is a strong rationale for targeting these particular isoforms, because they are the mutated disruptive channel that's in the brain. So digressing a little bit, I told you that 1.6 is also expressed in inhibitory neurons, and most of the work that's been done is to really understand what excitatory neurons do or change, how they change when this mutation is expressed in them. But it's also expressed in inhibitory neurons. And at first, the rationale was, well, if you've got a gain of function in inhibitory neurons, surely that would increase inhibitory drive and prevent these excitatory neurons from going crazy and generating seizures. So I had a very talented grad student, Eric Wenger, who's now at Ethan Goldberg's lab at CHOP, who worked on this project in my lab. And what he found was, yes, initially there was this hyperexcitability in these inhibitory neurons, but they were unable to sustain their firing. So this gain of function mutation in inhibitory neurons led to depolarization block, which means they're no longer able to fire action potentials, and they were significantly different in their firing rates compared to excitatory. What's the significance of this? Well, he was able to patch two neurons at the same time. He patched an excitatory neuron and he patched an inhibitory neuron, the SST in this case. And when he put on solutions that made the system epileptic by changing the magnesium concentration and adding 4-AP, you could make neurons fire epileptic bursts. And he found that the SST neurons started to fire much earlier, and that then encouraged the pyramidal excitatory neurons. But really, the pyramidal neurons started bursting when these inhibitory neurons went into block. So as soon as they started firing and went into depolarization block, that's when we saw the excitatory neuron burst. And the idea is that we believe that the inhibitory neurons are failing and the excitatory neurons are becoming hyperexcitable, and that's how the seizures are generating. So we have to really rethink of who's important here. Is it the excitatory neuron or the inhibitory neuron? To further address that question, we ended up using chemogenetic activation of SST neurons in wild type. So these are wild type mice. They don't have the gain of function mutation. What we did, we expressed DREAD receptors that are activated by CNO, and they are GQ coupled, and therefore you get an increase in excitation. When we did that in wild type mice, we were able to generate seizures just by expressing the chemogenetic GQ in SST. So if you drive SSTs to hyperexcitable state, you will have a seizure. You will make that mouse have a seizure. We recorded from these neurons to make sure that we were able to make them overactive. And we also found that these neurons went into block. So we believe that inhibitory neurons that express this mutation go into block, and that could be another mechanism by which these patients and these mice have seizures. So again, hyperexcitable output is encouraged or enhanced by the lack of inhibition because these inhibitory neurons fail. So just quickly, are there any clinical cases for loss of function of A-T? I've talked mainly about gain of function, and the answer is yes. There are quite a few patients out there that have a loss of function. They typically don't have epilepsy. Now, we're finding more and more of these patients that have absence seizures. That's when a patient is looking at you and then stops and just stares for a little while, and then will continue the conversation and not be aware that they had a seizure. That's an absence. But there are many patients that have loss of function that don't have seizures. What they do have is intellectual disability, and that's how they really get picked up. Parents will say, this is our third child, and this child was just not developing. They thought it was the child had autism, and then the patients or the pediatric neurologist will do a screen, and this is on the screen for intellectual disability, and it turns out a loss of function. We've recorded some of these from some of these patients, and you can see clearly you get current in wild type, but when you make these mutations, you no longer get any macroscopic current at all. Some of these channels make it to the membrane, that's in this Western blot plot, and they're just not functioning. Others just don't make it to the membrane at all, and that's due to the quality control machinery that we have. So summary of part one is gain of function mutations of 1.6 cause seizures, developmental delays, and motor problems. Mutations are de novo, so typically not inherited. Currently available AEDs are effective in some patients, but not all. The mouse model, the SCN8A 1768D mouse model and the 1872 mouse model exhibits many of the clinical phenotypes experienced by the patient, including seizures, motor problems, and most importantly, SUDEP. The excitatory neurons are hyperexcitable, and they have pro-excitatory alterations in sodium channel physiology. And recent work has suggested that inhibitory neurons may also be very important in this disease as they become hypoexcitable. I want to digress just a little bit here and say, well, I've talked mainly about an orphan disease, SCN8A. It makes up 1% of the pediatric population. And so it's an advantage for many reasons. FDA approval is fast-tracked because it's an orphan disease. But the bigger picture is temporal lobe epilepsy. And we know that 1.6 actually plays a big role in seizures in patients with temporal lobe epilepsy. And work from my lab and others has shown that, again, with temporal lobe epilepsy, and you can make animals have temporal lobe epilepsy in many ways. We use a hippocampal stimulation model. And when we do this, and mice or rats have spontaneous seizures, neurons become hyperexcitable. And studies from my lab have shown that the expression levels of 1.6 and 1.2 are upregulated, not only in the soma, but for 1.6 at the axon initial segment. And so here's another image from a paper a few years back that shows 1.6 expression at the axon initial segment is much more enhanced. And so if you develop a small molecule for 8A, you're going to be able to treat patients in that much larger 30% therapy resistant, and also the focal seizure group, because sodium channel blockers are the first line treatment for temporal lobe epilepsy, but also for focal seizures. And then 60% of patients with epilepsy have focal or partial seizures. And further evidence comes from my lab where we've used a flocked 1.6 mouse, so a Cree. So whenever we put Cree into this mouse, we can reduce or knock out 1.6. And here are just 3 animals where we gave them temporal lobe epilepsy, we made them have spontaneous seizures, and these are having spontaneous seizures, and we injected Cree at a critical time point, and you can see that we stopped the seizures in all 3 of these. We've repeated this 7 times now, and we see the same thing. And we targeted the hippocampus because the hippocampus because this is a temporal lobe epilepsy model. If you do the same experiment with GFP, we don't have an effect. So this is clearly a strong rationale for targeting 1.6 for temporal lobe epilepsy. So a summary of part 2 is epileptic neurons are hyperexcitable. The 1.6 isoform is upregulating epileptic neurons from temporal lobe epilepsy. And if you knock down 1.6, you can suppress seizures. So again, it provides a nice rationale for making subtype selective blockers, or at least CNS selective blockers for the treatment of epilepsy. So how do we treat these patients? Well, typically it's sodium channel blockers right now. They are non-selective, which means that they hit every single isoform. These are commonly used right now. If you talk to a pediatric neurologist who specializes in treating these patients, they will say we have some success with lamotrigine, carbamazepine, oxcarb, phenytoin, very high doses of phenytoin, unfortunately, which is also associated with side effects. But if you talk to some of these parents, they prefer their child to suffer some of the side effects and not have the seizures. But here you can see from a paper from way back, all of these drugs associated with toxicity because their TD50s are really, really low. And so you have a very small therapeutic window to treat these patients. My mouse model actually has audiogenic seizures. So we have a beautiful model where we can play a sound and they can have a seizure on demand. So we can inject drugs into them and then test them to see if it has an anticonvulsant effect. So it's a rapid screening model. And we've tested phenytoin, lamotrigine, and oxcarb. And sure enough, it does work in suppressing these seizures. But again, if you look at where the IC50 is compared to where the TD50 is, this is the problem that physicians have. They can only go so far. So what are other folks doing? Well, you may have heard of a company called Praxis, and they originally started off with Prax 330, which was known as it's a Gilead compound, GS967. GS967 was developed to treat cardiac arrhythmias, long QT3 syndrome, but it crosses the blood-brain barrier, and that program was dropped by Gilead, but Praxis have picked up 330, and we've done some work with them where we can show that this compound actually does block sodium channel current, blocks the persistent current as well, and because of that characteristic can normalize firing. So here's a neuron from a D+ animal, and you can see they have these abnormal action potential morphologies, these bursting, and in a dose-dependent manner, you can see that you can almost normalize them to what wild type will be. And when we tried a high dose of the compound on wild type neurons, we really didn't see much of an effect, suggesting that these compounds are highly selected for the inactivated state of the channel, which we know is much higher in these gain-of-function mutation patients. And if you feed the chow with this compound to the mice, you can see that you can prolong their survival. And as soon as you withdraw, obviously you follow a same pattern, and that's because you're not treating the underlying condition. You're just treating the sodium channel, and the underlying mechanism is still there. The mutation is still there. But this is very promising, and I know that Praxis have got other compounds. 562, I believe, is their lead compound that they are developing, and I think they've completed phase 1 clinical trials. So, but the Praxis compound is non-selective, and one of the problems with that is that we need selective and cardiovascular sparing compounds, because these are just some of the compounds that are out there that are non-specific. And the FDA in 2021 released a warning of cardiac arrhythmias, and they released the warning with lamotrigine. But as physicians will say, we shouldn't assume that lamotrigine stands alone among sodium channel blockers in terms of cardiac risk. So any non-selective, non-cardiac sparing compound is likely to have some kind of cardiac abnormality in the future. So the one biotech company that is moving forward with a selective blocker is Neurocrine, and so it was Xenon 901. Xenon developed this compound, Neurocrine had licensed it, and they are now, I believe, in starting phase 2 clinical trials. But clearly their compound, I think this is just one of their compounds, it's a sulfonamide, and it has high selectivity for the 1.6, as you can see here, compared to the others. Xenon also tested it on various mutations, because you want to know that it works on other mutations. These are clones that were developed, so these are sodium channel recordings, and you can see that it works very well on a number of these patient mutations. Is less effective on this 1617Q, which is one of the hotspots, And it's likely because that's where their compound binds, or it binds close to it. So this particular mutation disrupts that binding. So there's a lot of hope out there to make subtype selective blockers. The other avenue is genetic. So there is some work done by some biotech companies trying to target the 8A with antisense oligonucleotides. And Miriam Meisler has been doing a lot of preclinical work on this, and they have a ASO that I believe was developed by Ionis initially. I'm not sure if it's still being pursued by them. Sorry, wrongly. But what you can clearly see is when you inject these mice, and this is the 1872W mouse that if you express the mutation globally dies around about P18. If you inject the mice at P2 with this ASO, you can actually, in a dose-dependent manner, increase the survival. Miriam wasn't able to record EEGs because that's not what they do. But one assumes that during this time, the mouse was probably not having any seizures, but as soon as the ASO was no longer present, it obviously started to have seizures and die. And if they inject at P30, again, with another dose, you can see you can extend it. The problem here is that, you really don't want to be continually injecting humans with this ASO. So, I'm not sure how far ASO for 8A will go in its current state. So to finish up, summary of part 3 is that gain-of-function 8A mutations are best treated with sodium channel blockers. Unlike Gervais syndrome, which is typically loss of function, you do not want to give them a sodium channel blocker because you'll make it worse. Gain-of-functions are treated relatively well, but they're not well tolerated and could lead to cardiac abnormalities. So there is a real serious need to develop subtype selective blockers or genetic therapies for this treatment. So with that, I'd just like to thank my lab and the CUTE Foundation for all their support, and the NIH and the Epilepsy Foundation for funding. So I think we are able to take some questions now. Thank you.
Thanks very much indeed, Manoj, for your presentation. As you say, we're now very happy to open up the session for questions and answers. Just as a quick reminder, questions should be sent in through the control panel, so you can type those in there at any point. And at this point, I'd like to bring back in Eddie, who's going to act as our compere. Eddie, you're going to hopefully pose the questions that come in from our audience this evening.
Yeah. Right. Okay. Well, fantastic. Thanks, Manoj, for a great presentation, and thanks giving it a drug discovery tilt for us industry people in the audience. So yeah. So the first question is, so what do you think the persistent current is? Because from your N1768D, you're getting bigger window currents with bigger persistent currents. Is that what you believe? Do you think the window current is driving the persistent current, or it's another mechanism?
I think the window current is, because the fact that your shift in the steady state inactivation is related to the persistent current. It's complicated because one thing that's missing is really good single channel characteristics of this mutation. So if we were able to do some more single channel recordings, we'd get a better idea of, do we get lots of sweeps with delayed openings that open much later on? So I think they are related. And I think that's probably the best answer for that. I think that's probably why you're getting this persistent current. But then you have this population of the channels that are just never going to turn off. Right.
Okay. And what about, yeah, and the depolarizing block with the gain of function is interesting. But is that specific to NaV1.6? What about the other sodium channel subtype related epilepsy? Is there any evidence there that gain of function mutations inhibit interneuron activity?
So inhibitory neurons tend to express mainly 1-1, and so the Gervais syndrome is, there are some Gervais patients that have gain of function NaV1.1, but no one's, I don't believe anyone's really recorded from neurons from mice, because I don't think we have a mouse model for it, where they would probably see the same thing, right? So in Gervais, you have a loss of function that tends to give you Gervais syndrome because those neurons don't fire anymore. But in those patients that have Gervais because of a gain of function in 1-1, I bet you're seeing the same thing. So 1-2 doesn't seem to be heavily involved in controlling excitability of the inhibitory neurons, but 1-6 does. And I think that in this situation, we're really seeing I mean, you could argue and say, are we really seeing Gervais in 8A? Is it really an inhibitory neuron problem? And we just don't know that until we do some in vivo recordings.
Right, okay. And I've got another question about the genetics, which is there an association with pain as well with genetics? Do those patients have a dysfunction of pain?
So that's a great question. I've asked the parents often, I always ask them this question: does your child have hypersensitivity to pain? And it turns out that they claim, and they're right. I mean, you really want to ask parents these questions and not physicians. They will say that they seem to have a high tolerance for pain. So they will say they have other children who, when they fall over, will hurt themselves and cry, and their child with 8A gain of function typically falls over and just gets up and keeps going. And it's really interesting because I would have thought it'd be the other way around. Now, I just don't know, because they have this gain of function 1/6 in their dorsal root ganglion neurons and in the spinal cord, whether there's some kind of desensitization going on. I don't know, whether there's some kind of plasticity that's made them almost analgesic, right? So yeah, that's a great question.
There's been no nerve conduction studies or anything like that on these patients.
No, and you know, we actually have the mice. So one thing we're thinking of doing is to do some pain studies on our mice, because that would be very interesting.
Right. Okay. And what about mechanisms of block? So what's your, because you talked about the different approaches, either non-selectively blocking persistent current, or specifically targeting that 1/6, which would tend to be the peak current rather than a persistent component of 1/6. So what's your take on that? What do you think is the better strategy, or your preferred strategy?
Well, the preferred strategy would be to have I think like the Neurocrine compound, which I think has a very strong block of, affinity for the inactivated state and the 1/6. So I think, all of these sodium channel blockers that are out there, the non-selective ones, will have affinity for the inactivated state, which is why they're somewhat affected at blocking the persistent current, and then having a smaller effect on the transient current, which is where your side effects come from, because they're doing it to every channel. But I think that if you have a 1/6 selective blocker that has a high affinity for the inactivated state and that particular isoform, because this mutation, kind of enters that inactivated state more readily, or is in that state more readily, your compound's going to take that out. So it's I really think that that's what you really want to treat these patients. Now, at the same time, I will say it's going to be a titration issue, because 1/6 is critical. Okay, if you block 1/6, it's lethal. So so every patient's going to have to be titrated carefully to ensure that you're not blocking too much of the transient current.
Right. Okay. Yeah. Well, does that mean that the ultimate would be a selective NaV 1.6 persistent current, which seems, sort of out of reach?
It seems out of reach, but if we can get a better idea of what is the gate, what is that slow inactivation gate looking like, or that kind of confirmation looking like, and can we then specifically target when they get into those distant inactivated states, right? That we can just have a much higher affinity there, so that we're less likely to affect the overall transient current. But it is equilibrium, as you mentioned before, and so you'll shift that equilibrium, but hopefully towards less persistent.
Right. Yeah. Yeah. What about for the TLE patients? I mean, it's the advantage of the genetics is the understanding of the disease it's more homogeneous, but I would have thought TLE it isn't so. So are you sure that sodium channel block is always going to relieve symptoms, or could it exacerbate?
So I think we know it does relieve symptoms, because that's more or less what patients are treated with, is typically sodium channel blockers for temporal lobe epilepsy. Work from my lab and other labs have shown that the biophysical properties of the sodium channel from these TLE neurons is different. So actually the steady state inactivation is shifted as well. And we know that's not because of a mutation, that's post-translational modification, right? So these channels are now behaving just like the gain of function mutations, to be honest with you. And so again, their inactivation gating is also changing. We're lacking the single channel recording from these neurons, so that we can better understand what's changing in the biophysical properties of these single channels. But again, that's why I think, in a way, 8A and temporal lobe epilepsy have a lot in common. And I think if you can get a compound that works well in the orphan disease, I think it's going to open up the market for them.
Right. Okay. Yeah.
Patients.
Okay, and what about the resurgent currents? Is that because that's sort of interesting for 1.6? Is I mean what because obviously you showed that the persistent current blockers that they removed the resurgent current because basically you prevented threshold. But is that another avenue? What does it tell you about epilepsy, the resurgent current? The fact that you saw a clear increase in in the amplitude.
It's interesting. I don't know if I have an answer for that, but what I would tell you is that if a neuron wants to fire, that these epileptic neurons believe that that's their normal state. Okay, when they're firing at those high levels and producing seizures in temporal lobe or 8/8, that's where they think that's normal. And so I believe that whether it's the mutant channel expressed in these patients that's giving you the larger persistent current, or the wild type also now giving you a larger resurgent current because of post-translation modification, the neuron is trying to get you there. And so I think they're somewhat related to the gain of function mutation, because clearly the DDs had it larger and they don't have a normal wild type channel. But I think the other wild type channel is probably also being changed to allow that neuron to fire. So I guess my point is there's so much plasticity that goes on because you've reset what I would say the thermostat for excitation of these neurons, and they'll do anything they can, including probably increasing K channel expression. Now, I'll tell you, in our mice, this has been done, and K channel expression does not change, but in epilepsy, it will. And again, it's just to encourage excitation.
Yeah.
So I'll put a question to you. You could theoretically treat these patients with a K channel opener, and so that's something that's being looked at, right? Because you just want to increase repolarization.
Sure. Yeah. Well, have patients ever taken retigabin? Has that been trialed?
No, they haven't. As far as I know, no. I know that Xenon are developing K-channel openers for treatment of epilepsy. I believe they just licensed one for partial seizures, I think, or some type of seizure. But I will tell you one interesting fact, and that is Keppra, which is this SV2A, is levetiracetam, is very dangerous for these patients. For some reason, it sends them into status. And we're trying to get the word out to pediatric neurologists that when you see an 8/8 patient, please do not give them Keppra, because typically Keppra works really well in temporal lobe epilepsy. So that's usually the first line of treatment for these kids, and they end up in the ER having status. And some have actually died. We have no idea why Keppra makes their seizures much worse. But clearly there's something different going on with these patients compared to other patients.
All right, fantastic. So one question is there are people pursuing a structure-based design for NAV 1.6? Is there a crystal structure, especially an inactivated state structure?
Yeah, so I think the crystal structure we have is for the bacterial channel, and so I think people are using programs to try and develop their molecules, but it's difficult because it's the bacterial channel. The bacterial channel is one domain that comes together, and it's structurally different. And again, unless we really know what this inactivated state is, because that's what we want to target, I think it makes it difficult. But I am actually not an expert on how far people have gone with these in silico models. I know people use them. We have basically used our structure activity relationship data from ion channel screening and using them in the mice to develop our compounds.
Okay, brilliant. Okay, well, I think we'll stop there. So thanks very much, Manoj, for answering all those questions. Okay, so this is well, thanks to everyone for joining our webinar today. So this is actually the last Metrion webinar of the year, but we'll be running more webinars next year. So we'll be announcing the speakers in the new year. So yep, thanks for everyone for joining, and I hope you enjoyed the talk. Thanks very much.
Thank you.